DNA replication. First question. DNA replication requires. First option, DNA polymerase only. Second option, DNA polymerase and ligase. Third option, ligase only. Fourth option, RNA polymerase. DNA replication, one DNA molecule that produces two daughter DNA molecules. In this, it is the mother DNA. Two strands, they are separate during the replication. Two daughter DNA molecules are formed. It is the old strand and it is the new strand. It is the old strand and it is the new strand. So it is the DNA replication is semi-conservative method. DNA replication is semi-conservative method. Now, this DNA, the two strands of DNA that separate from one another in particular region only, that is origin of replication. Both strands act as template. One strand 3-5 polarity, that is leading strand. Another strand 5-3 polarity, lagging strand. New strand always synthesized in 5-3 polarity only. Now, in 3-5, new strand that continuously synthesizes without any breaking. But in this one, wherever it separates, fragments of DNA that is formed. For the formation of the new strand, nucleotides that are attached. For the attachment of nucleotides, DNA polymerase that is essential. So, DNA polymerase that polymerize addition of the nucleotide, deoxyribonucleotides. So, in one strand, that is a 3-5 polarity DNA template that required only DNA polymerase. But here, fragments are the pieces of DNA form. They are called Okazaki fragments. After their formation, for the attachment of these fragments required DNA ligase. So, strand that shows the 5-3 polarity required both DNA polymerase and DNA ligase. So, for the replication of DNA, these two enzymes that are essential are necessary. They are DNA polymerase that is required for the polymerization of the deoxyribonucleotides and DNA ligase that is required for the adding of fragment. They are Okazaki fragment in 5-3 polarity. So, the correct option is the 2. Both DNA polymerase and DNA ligase required. RNA polymerase not at all involved in DNA replication. RNA polymerase not at all required in DNA replication. For the first question, answer is the 2. Both DNA polymerase and DNA ligase is essential for DNA replication. Question number 2. Okazaki fragments are synthesized on option 1, leading strands of DNA only, lagging strands of DNA only, both leading and lagging strands, and fourth one, complementary DNA strand. Two strands of DNA when separated from one another in the origin of replication. The strand 3-5 polarity is called leading strand. And another strand that shows 5-3 polarity, that is lagging strand. the template leading strand in this new DNA strand that synthesized continuously into 5-2-3-1 polarity. But on the lagging strand, new DNA that synthesizes in pieces are the fragments. Those fragments are called Okazaki fragments. So Okazaki fragments that are formed on lagging strand, not on leading strand. So in leading strand, continuous DNA synthesizes new DNA strand. On lagging strand, discontinuously new DNA strand that synthesizes. Those discontinuous DNA fragments called Okazaki fragments. So Okazaki fragments that are formed on lagging strand only, not on leading strand. So the answer is 2. Question number 3. In which of the following DNA not directly involved? In which of the following DNA not directly involved? Option 1, replication. Option 2, transcription. Answer 3, translation. Answer 4, transformation. Replication of DNA. Both strands are involved in the DNA involved in the replication. Transcription is the DNA strand from the DNA strand, RNA that synthesizes, that is called transcription. That process called transcription. Translation, based on the information present on the mRNA, it is mRNA, polypeptide chain is formed. So it is the polypeptide chain. It is the mRNA. Transformation, it is the process of the transfer of DNA from one bacterium to other bacterium through medium. It is the bacterium and it is the bacterium. After the death, it comes out and incorporated with DNA of another bacterium. So in these 4 options, in replication DNA involved, in transcription DNA involved, in transformation also DNA involved, but in the translation DNA not at all involved, mRNA involved. In translation mRNA is involved. Formation of polypeptide chain in accordance to nitrogen base sequence or the genetic information present on the mRNA, that is called the translation. In translation only, DNA not at all involved. So answer is 3. Question number 4. It is assertion reason question. Assertion. DNA replication does not occur in interkinesis. Reason. In meiosis, nuclear division and chromosomal division occur each once. Option 1. Assertion and reason both are true and reason is correct explanation of assertion. Option 2. Assertion and reason both are true but our reason is not correct explanation of A. Option 3. Assertion is true and reason is false. Option 4. Assertion is false and reason is true. Meiosis completes in 2 steps. That is meiosis 1 and meiosis 2. Before meiosis 1 and after meiosis 1, interphase occurs. So it is the interphase 1. Next, meiosis 1. Next, interphase 2. Then meiosis II. The interphase II that present in between meiosis I and meiosis II called interkinesis. Interphase II that present in between meiosis I and meiosis II, that is also called interkinesis. DNA replication occurs in interphase I only. But in interphase II or interkinesis, G1 and G2 phases only occur. S phase is absent. In interphase I, G1 phase, next S phase, and then G2 phases. All the three phases present, but in interkinesis, only G1 and G2 phases present, S phase is absent. So, overall in this meiosis, only once DNA replicates. DNA replication occurs only once. And karyokinesis, that is the division of the nucleus that occurs in meiosis I and meiosis II. So, answer, assertion correct, reason is the wrong. Both DNA replication and karyokinesis occur in meiosis I and meiosis II, that is wrong. DNA replication occurs only once, that is just before meiosis I. So, answer is 3. Question number 5. Study the following and identify the correct statements. Statement 1. DNA dependent DNA polymerase catalyzes the polymerization of nucleotides always in 5 to 3 direction. The replication is continuous on the template with the polarity 5 to 3. Replication begins at the replication fork. Fourth statement. In lagging stand, synthesis of all Okazaki fragments is regulated by a single anchor. Option 1, 4th and 3 statements. Option 2, first, second and third statements. Option 3, first, second and fourth statement. Option 4, second, third and fourth statements. The two strands of DNA separate from one another at origin of replication. Both strands act as template in DNA replication. DNA dependent DNA polymerase. DNA dependent DNA polymerase that add nucleotides in 5 to 3 end polarity. 5 to 3 end polarity. Here also 5 to 3 end polarity. Now, on 3 to 5 template, the synthesis is continuous. On 5 to 3 polarity stand, it is discontinuous. It is discontinuous. So, when these two strands separated, DNA strands separated, that looks like a fork, that is called replication fork. In the origin of replication, two strands they are separated, looks like replication fork. So, during this one, it is a leading strand. It is a lagging strand. RNA primer is required for the attachment of DNA dependent DNA polymerase. So, only one anchor that is sufficient toward the leading strand. But during the formation of new DNA strand on lagging strand, each Okazaki fragment required one anchor. There is one RNA primer. So, when DNA new strand that synthesizes on the 5 to 3 polarity, number of RNA primers or the anchors that is equal to number of Okazaki fragments. So, correct answer 2. That means First statement, second statement, third statements, the 3 right, and fourth statement, that is wrong. The lagging strand synthesis of all Okazaki fragments is regulated by single anchor, that is wrong. Many anchors that are required, number of anchors required for the synthesis of new strand on lagging strand, that is equal to number of Okazaki fragments. Question number 6. Arrange the following steps in sequential order with reference to the experimental proof of semi-conservative method of DNA replication in Escherichia coli by Messelson and Stahl. A. Culturing of E. coli bacteria in heavy isotope of N15 contain ammonium chloride medium. B. Incorporation of heavy nitrogen into newly synthesized DNA and other molecules of E. coli. C. Transfer of cells into the normal N14 contain ammonium chloride medium. D. Separation of various samples on cesium chloride gradients to measure the densities of DNA of N14 and N15 types. Option 1, B, C, D, A. Option 2, C, A, B, D. Option 3, A, B, C, D. Option 4, D, A, B, C. Messelson and Stahl first cultured E. coli, Escherichia coli in the medium with N15 contain ammonium chloride. So N15 that entered into the newly formed bacterial DNA and other nitrogen containing molecules. There are the proteins also. Nitrogen that present the nucleic acids and the proteins. So nitrogen N15 that incorporated in nucleic acid, the DNA, RNA and the proteins also. Then the bacteria are shifted to the another medium which consists N14 contain ammonium chloride, and then different durations separated and measure in the cesium chloride solution gradient. So it is a sequence of various steps in the experimental proof of semi-conservative DNA, semi-conservative method of the DNA replication. So first, when it was taken NH4Cl, N15 contain ammonium chloride, all are heavy. N15 contain ammonium chloride, all are heavy. And then when shifted to this one, it is a heavy and this is the hybrid. So sequence of the various steps in the experimental proof of the DNA replication semi-conservative method of DNA replication. First, culturing of the bacterium on medium with N15 contain ammonium chloride and N15 that incorporated in nucleic acids and proteins, that means the nitrogen containing all molecules. Then shifted to the another medium which consists the N14 contain ammonium chloride containing medium, and then subjected to centrifugation with the help of in the medium cesium chloride. So correct answer 3, A, B, C, D. Question number 7. In a double-stranded DNA molecule, both the strands were labeled with a heavy isotope N15 and was transferred into N14 medium. It undergoes replication for 3 generations. What is the ratio of DNA molecules with both the strands with N14 and N14 plus N15 hybrids? Option 1, 1:1. Option 2, 1:3. Option 3, 1:2. Option 4, 3:1. It is the parental generation. Both strands are labeled with N15 and then allowed replication on N14 containing medium. In the first generation, it is the N15, newly stand N14. It is the first generation. In the second generation, 4 DNA molecules are formed. These two are separated. It is the only N15. It is the only N15. Remaining all strands are N14. That means N15, N14. It is the hybrid. And these two are the light and it is the hybrid. So it is the second generation. In the third generation, from 4, 8 are formed. So number of DNA molecules formed in n number of generation, that is the 2 to the power of n. N is equal to number of generations. So 3 generations, 2 to the power of 3, that is 8. So at the end of third generation, 8 DNA molecules are formed in any generation except parental generation. In any other generation, N15, N14 are the hybrids are always 2. Hybrids are always 2. So out of 8, 2 are hybrid and remaining 6 are light. So the question, that is the ratio between the N14 contain DNA molecules and both N15 and N14 contain DNA molecules. That is the 6 is to 2. That is the ratio 3 is to 1. So 6, they consist of only N14 and these two consist both N15 plus N14. The ratio between both strands with N14 and DNA molecules with both N15 and N14, 6 is to 2, ratio 3 is to 1. So answer is 4. Question number 8. Identify the correct match among the following. Column 1, option 1, Wachawa's enzyme. Column 2, DNA dependent DNA polymerase. Second option, 3 to 5' end, template strand of. Column 2, leading strand of DNA. Option 3, 5 to 3, template strand of DNA. Acts as template strand of hnRNA, heterogeneous nuclear RNA. Option 4, transcriptase in prokaryotes, transcribes only mRNA. Wachawa's enzyme, that is useful for the production of RNA with defined sequences. So enzyme DNA dependent DNA polymerase not involved. Wachawa's enzyme is not the DNA dependent DNA polymerase. That is DNA independent RNA polymerase. Second option, 3 to 5' end template strand of, leading strand of DNA. Leading strand of DNA. So during the replication of DNA, one strand 3 to 5' end, another strand 5 to 3' end polarity. It is a leading strand. and it is the lagging strand. So in this one, new DNA that synthesizes continuously. So it is the correct one. And third option, 5 to 3 prime end template strand of DNA acts as the template of strand of hnRNA. That is transcription. In transcription, only one strand of DNA acts as a template. That is the 3 prime end to 5 prime end template of DNA only acts as template. 5 to 3 prime end strand not involved in transcription. So third statement wrong. And fourth option, that is transcription in prokaryotes transcribes only mRNA. That is also wrong. Transcriptase. In case of the prokaryotes, all types of RNA, mRNA, tRNA, ribosomal RNA, all are synthesized. Transcriptase, that synthesize all types of, not only mRNA. So fourth option also wrong. The correct answer, 3, 5 and template strand, leading strand of DNA. So correct answer is 2.